Chapter 1 Sexual Reproduction in Flowering Plants, Short Notes and Summary from Class 12th, Subject Biology. Introduction. Reproduction is essential for continuation of species. Sexual reproduction in plants creates genetic variation, giving adaptive advantage. Flowers are the main reproductive structures in angiosperms. Sexual reproduction in angiosperms involves pre-fertilization events, gametogenesis and pollination, then fertilization, then post-fertilization events, seed and fruit formation. 1.1 Flower, a fascinating organ of angiosperms. Flower, modified shoot, specialized for reproduction. Halls of a typical flower: first calyx, sepals, protection in bud stage; second corolla, petals, attraction for pollinators; third androecium, stamens, male reproductive part; fourth gynosium, carpels, female reproductive part. Bisexual flowers have both androecium and gynosium, example hibiscus. Unisexual flowers have either androecium or gynosium, example papaya, watermelon. 1.2 Pre-fertilization, structures and events. Pre-fertilization includes gametogenesis, formation of gametes and transfer of gametes. 1.2.1 Stamen, Microsporangium and pollen grain. Stamen, male reproductive unit, consists of anther, produces pollen grains; filament, stalk supporting anther; anther structure, typically bilobed. Each lobe has 2 microsporangia, total 4 microsporangia. Epidermis, endothecium, middle layers, tapetum, nutritive, binucleate cells. Microsporangium contains microspore mother cells, MMC. Microsporogenesis, formation of microspores haploid from pollen mother cell PMC by meiosis, microspore tetrads. Pollen grains, male gametophytes, wall layers, exine, sporopollenin, resistant, germ pores, and intine, cellulose, pectin. Cytoplasm contains vegetative cell, large, food rich, and generative cell, forms 2 male gametes. Shed either at 2-celled 60% angiosperms or 3-celled stage. Allergies: some pollen, example Parthenium, cause asthma, bronchitis. Viability varies. Rice, wheat, approximately 30 minutes. Rosaceae, Leguminosae, Solanaceae, months. Stored in liquid nitrogen, -196 degrees Celsius in pollen banks. 1.2.2 The pistil, Megasporangium, ovule and embryo sac. Gynosium, single or many pistils. Monocarpellary, 1 carpel. Multicarpellary, many carpels. Syncarpus, fused. Apocarpus, free. Pistil parts: stigma, pollen landing; style, slender stalk; ovary, contains ovules; ovule, Megasporangium; panicle, stalk; hilum, junction; integuments, coverings; micropyle, opening; chalaza, base; nucellus, nutrition; embryo sac. Megasporogenesis, megaspore mother cell MMC diploid to meiosis to 4 haploid megaspores. 3 degenerate, 1 functional megaspore develops into embryo sac. Embryo sac, female gametophyte. Development is monosporic, form 1 megaspore. Mature sac, 7-celled 8 nucleate. Micropylar end, egg apparatus, 2 synergids plus 1 egg cell. Chalazal end, 3 antipodals. Central cell, 2 polar nuclei. 1.2.3 Pollination. Pollination, transfer of pollen from anther to stigma. Types of pollination: autogamy, same flower, rare, requires synchrony. Clistogamous flowers, never open, example viola, oxalis, always self-pollinated. Geitonogamy, different flower, same plant, functionally cross-pollination, genetically self-pollination. Xenogamy, different plant, true cross-pollination. Agents of pollination, abiotic, wind, light, non-sticky pollen, feathery stigma, example grasses, maize. Water, rare, 30 genera, example Vallisneria, Hydrilla, Zostera. Biotic, insects, bees, butterflies, birds, hummingbird, bats, primates, reptiles. Outbreeding devices, different timing of pollen release and stigma receptivity, different positions of anther and stigma, self-incompatibility, genetic rejection of self-pollen, unisexual flowers, monarchy, diarchy, pollen-pistil interaction, stigma checks compatibility, promotes, inhibits pollen germination, artificial hybridization, emasculation, removing anthers before they mature, bagging, covering stigma to prevent unwanted pollen. 1.3 Double fertilization, pollen tube enters ovule via micropyle, enters synergate via filiform apparatus, releases two male gametes, one gamete plus egg, syngamy, zygote 2n, other gamete plus two polar nuclei, triple fusion, primary endosperm nucleus 3n. Since syngamy plus triple fusion occur, the process is called double fertilization, unique to angiosperms. 1.4 Post-fertilization, structures and events. 1.4.1 Endosperm. Endosperm development precedes embryo, provides nutrition. From primary endosperm nucleus 3n. Types, free nuclear, most common, example coconut water, free nuclei, kernel, cellular endosperm. Cellular, fate, consumed by embryo, non-endospermic seeds, tea, groundnut. Persist, endospermic seeds, castor, coconut, cereals. 1.4.2 Embryo, formed from zygote at micropylar end, stages, proembryo, globular, then heart-shaped, then mature embryo. Dicot embryo, two cotyledons, epicotyl with plumule, hypocotyl with radicle and root cap. Monocot embryo, one cotyledon, scutellum, polyptile, shoot sheath, coleoriza, root sheath. 1.4.3 Seed, final product of sexual reproduction, parts, seed coats, cotyledons, embryo axis. Albuminous seeds retain endosperm, example maize, wheat, castor. Non-albuminous seeds, endosperm fully consumed, example pea, groundnut. Perisperm, persistent nucellus, example black pepper, beet. Fruit formation, ovary to fruit, ovules to seeds. True fruits from ovary only. False fruits with thalamus, example apple, strawberry. Parthenocarpic fruits without fertilization, example banana, induced by hormones. Advantages of seeds, dispersal, dormancy, food reserves, protection, variation. Seeds viability varies months to thousands of years, example 10,000-year-old Lupinus arcticus seed germinated. 1.5 Apomixis and polyembryony. Apomixis, seed formation without fertilization, asexual reproduction mimicking sexual, seen in Asteraceae and grasses. Embryo develops from diploid egg without meiosis or nuclear cells, example citrus, mango. Useful in agriculture, hybrid seeds can be maintained without repurchasing. Polyembryony, more than one embryo in a seed, common in citrus, mango, can arise from nuclear cells or zygotic embryo. Summary: flowers, reproductive organs in angiosperms, androecium, pollen grains, male gametophyte, gynoecium, ovules, female gametophyte, embryo sac, pollination by abiotic, wind, water, or biotic, animals, agents, double fertilization, syngamy, zygote 2n plus triple fusion, endosperm 3n. Post-fertilization events, endosperm nutrition to embryo new plant, seed final product, fruit from ovary. APOMIXIS, seed without fertilization, polyembryony, multiple embryos in one seed. So friends, we have completed short notes and summary of chapter 1, sexual reproduction in flowering plants. We have already studied the question answers of this chapter. You can find the link in the description and on the screen. If you like the video, kindly like, hype the video and share it with your friends. Also subscribe to my channel for more such videos. We will meet in the next video. Thank you for watching.